I see some familiar faces from from yesterday. Didn't have my coffee yet. Hi, I'm Sofia. That is, I'm Sofia Yaneska, and I will be your host here today. And yes, I would like to start with your introduction. So this is Vladimir Kovacevic. He's a senior algorithm developer at BGI Research Serbia. And spatial transcriptomics offers a deep dive into the cellular composition of tissues, providing high-resolution insights into gene expressions and cell positioning. Vladimir's talk will explore the challenges of processing vast data sets from millions of cells with thousands of genes, focusing on innovative algorithms for tasks like cell community detection and cell type annotation. So Vladimir, you can start.
Thank you. So the convention of the time traveling will be held yesterday. So thank you for the introduction. It's pleasure to be here. I come from the background of computer science, but for the last 9 years I've been working in bioinformatics and biotech domain, and this area through all these years became much fonder to me than my original education. So in this presentation, I would firstly like to bring you closer the entire technology of spatial transcriptomics. I want to present you how the data look like and what can it be done with it, what useful information could be extracted from it. And then in the second part, I would like to show you two algorithms that our team here in Belgrade in BGI Research have developed. So stay put. Spatial transcriptomics, it's been with us as a commercially available technology for more than 8 years now. In 2020 it was announced to be the method of the year, and through its process it allows us to sequence the thin slices of the tissue and obtain individual expressions of the individual cells. So you can cut the solid tissue, put it on the chip with those thin wells spots, and when you remove everything beside DNA genomics material, this genomic material, it falls into these spots, and you can sequence it there, and you can also keep the information about the position of the individual cells in the tissue. So when sequencing experiment is finished, you actually obtain gene expressions for individual cells. So this as a rows of this matrix, you can have thousands of cells. In some experiments, having hundreds of thousands of cells, and as the columns, you obtain gene expressions of the individual cells, like 30,000-40,000 of genes you obtain for every cell after reading RNA reads from the sequencing process. And as I said, for every cell you obtain X and Y coordinates in the tissue. So you can after this first part, upstream analysis, you can reconstruct the tissue and obtain, as I said, gene expression for every individual cell. So with that, what can be done? This is just one of the examples. So you can sequence, you can study many different organisms. Currently, the technology is mostly used in fundamental science, in obtaining knowledge and information about the function of the cells and inside of the tissue, how the cells are communicating, how the microenvironment in the cell is functioning. This is an example of sequencing of the mouse embryo from day 9 to day 16 with reconstructed cell types present in it. And through it you can actually see the organogenesis of the entire organism. So you can see how the organisms, how the organs are developing from early stages of the embryo to later stages. This sequencing was done in a stereo-seq technology developed by BGI. Of course, you're wondering how it's possible to obtain all of this from single organism. Well, it's not the same mice, of course. Every day they took a different one. Another example, what you can do. So the previous one was temporal dependence, and here you can obtain also spatially consecutive slices of the tissue, like what they did here. So 86 consecutive slices were sequenced, and we performed experiments on each of them and reconstructed cell types. So here you can see 3D reconstruction representation of the entire organism with the different cell types present in it. It represents valuable source of information and insight to the scientists about the functioning of the organism and the interaction inside of its cells. And in order to obtain such a beautiful insights, of course, you will need to process this data and process it a lot. So these are some groups of the algorithms that exist in the spatial transcriptomics. So firstly, what is usually done is some quality control, normalizations. Then you are able to obtain annotations of the cell types. You're able to calculate which genes are present in each of the clusters, in each of the cell types. You can identify them. You can perform much complex analysis, such as trajectories. You can see which cell type was derived from which, and how this process was done during the evolution of the cell. You can define and try to find the genes that are spatially variable, so they are expressed in some parts of the tissue and in some not. And based on that, you can find some regions of interest, some functional domains in the tissue, and you can perform 3D reconstruction. And in order to do all of this, you probably notice we're dealing with huge amounts of data. So we have hundreds of thousands of cells in the modern experiment, and for every cell we have 30,000-40,000 of genes. Even though this matrix is very sparse, so we don't have information on every spot, still we're dealing with huge amounts of data. And in order to process this, we would need very high computational resources, usually done in the cloud, and it's very convenient to have a cloud platform specially developed for this type of data, and also for other analysis, such as DCS cloud environment, also developed by BGI. It's a cloud-based platform for multi-omics data analysis, where users without computational knowledge are able to manage, analyze multi-omics data while keeping it safe, so they can collaborate, share, utilize many bioinformatics tools. Some of the algorithms that I mentioned in the previous slide are available there, and they can run computationally intensive tasks only in a few clicks. So the platform is equipped with a workflow description language developed by Broad Institute, so the analyses are all run inside of the Docker images, and they are reproducible. Also, the platform contains a large number of multi-omics tools and interactive analysis, so it's very convenient to do at least basic analysis and get some basic insights about the platform. So in the following part of the presentation, I would like to give you insights about two concrete algorithms. I will go technical, so I hope it will be interesting to you through some concrete examples to get insights about the possibilities and the real-time use cases. So as mentioned, some of the first tasks when obtaining information about the spatial transcriptomics data is to assign cell type to every cell in the tissue. So we want to see whether it's neuron, whether it's erythrocyte, lymphocyte, what features does it have. And the technology that was mostly done before spatial transcriptomics was invented, and also very popular technology nowadays, is single cell sequencing. where you can obtain gene expressions from individual cells without spatial coordinates. That's the main difference, let's say. And in that technology, the flow for obtaining cell types is usually done in these 3 steps. So when you have this cell gene matrix that I mentioned, you firstly would perform some unsupervised clustering to see which cells are similar to which and to group them together, to cluster it. Then for every cluster, you would find which genes are expressed in it and not expressed in the others. So which genes represent this cluster. And based on that genes, you would search existing databases of cell types to find the closest match. And you would assign this closest match to the cluster that you identified. And that's the flow for obtaining cell types in single cell RNA-seq sequencing. So, but for spatial transcriptomics, this flow, it doesn't work. Why it doesn't work? Because of the limitations of this technology, and the limitation is existence of gene dropout. So actually, in all of the spatial transcriptomic technologies, we are not able to obtain full landscape of gene expression for every cell. So we do not get all the genes in the cell. We get like 20–30% of it top, and the rest is just gone. So for the rest, we don't know whether it's 0 or we didn't capture it during the sequencing process. And because of that limitation, because of this gene dropout, we are not able to reliably find these marker genes. Because this artificial dropout that was done during the sequencing process can give us false information about the variability. So we could, the algorithms could be tricked for finding marker genes. Maybe they are not varying because of reality, because of their nature of the cell types that they belong to, but actually we didn't capture it correctly. Because of that, this is just an example comparison between number of genes in the cell comparing to single cell technologies and the spatial transcriptomics. So with single cell marked here with red, with these two samples belonging to different technologies, we can see that we have much higher number of genes inside of each cell. So in order to overcome this limitation, one of the solutions is to use high quality reference single cell data set from the same tissue. So for example, since nowadays many sequencing experiments were done, and a lot of laboratories, institutes, when they finish the experiments, they publish their data. So if we're analyzing data that already have available high quality single cell data sets for our tissue of interest, we can download it and we can use it as a reference and map our cells, our gene expressions of our cells, map it to find the closest match. So this would be single cell reference with the already obtained annotations as shown on the previous slide. And for every cell, we look for a closest match. And we can, in essence, compare these gene expression vectors to one in the reference data set and try to find which one should be assigned. Using this principle, we have developed CODI, the acronym is for contrastive distance, a tool for cell type annotation, which receives on its inputs the reference single cell dataset with cell type annotations and spatial transcriptomics dataset, test dataset that we want to annotate. And we perform in parallel two different tasks. So the first one uses statistical vector distances in order to find the closest match from the single cell reference. And in parallel, we perform training of the deep learning encoder in order to predict cell types. So we train deep learning encoder with single cell reference data, which have already annotations, and perform prediction on spatial transcriptomics data set. And as a final result, we will obtain cell types for every individual cell, together with the confidence map, telling us how reliably our tool predicted the cell type of each individual cell. So under the hood, this is the diagram that shows how the architecture of this encoder look like. First step is data augmentation. So since, as I showed these histograms on the 3 slides before, you can see that distributions of the data that we use for training and for inferring is different. So this, we call it augmentation, but it's actually some kind of data reduction, because we want to bring closer these two distributions, and we are doing it by randomly doing the dropping out, what was done during the sequencing process. So we would have similar number of genes in the cell during the process of training this deep learning encoder, like in the spatial transcriptomics data. And when we were selecting deep learning architecture, we gave us some thoughts and discussions what could be used, and of course the latest trend for large language models gave us information we should go for it. Everybody is using it. You know, the reviewers of the paper would love it. So no, we didn't go for it. For large language models, we used the contrastive learning based encoder, because in essence this is a simple problem, because we have gene expressions and we have annotations, and we should perform at the end multi-class classification. This is our task, and for it we wanted firstly to experiment with much simpler architectures. Smaller time of the training, and the entire process would take would be a lot easier to be done. So in essence, contrastive learning, it performs creation of the embeddings, while for every input training instance, the cells belonging to the same cell type are treated as positives, and their embeddings should be as similar as possible to the embeddings of the anchor. And cells belonging to other cell types are pronounced as negatives, and they should have embeddings which are much more different than cell type that is currently on the input. And as an output, we obtain these embeddings, which are fed to the classification head. And here we're dealing with two losses. So the first loss is done as a cosine distance, standard loss in contrastive learning, and the second one is a classification loss, which tells us how accurately our network guessed the correct annotation for the cell. So these two losses are accumulated with some configurable weight, and they are passed to the optimizer which performs its minimization. And as an output, we have prediction and probabilities for each cell type, which we can use to construct this confidence map that I showed you. This is just some closer insight how it looks like in the inside of the batch. So since we cannot put all the samples on GPU at the same time, so we divided it into batches, and inside of every batch we mark which cell types are the same, which are the different from the current one, and we can obtain this supervised loss. This would be the result. So after we obtain embeddings and project them in the two-dimensional UMAP space, you can see that different cell types are positioned differently in this space, and this is just an insight to see whether the task for the classifier that comes after it would be simple or difficult. So it can tell us some information whether if they are separated easily in this in this space, then the classifier shouldn't have tough task to do it afterwards. And on the right, you can see some trick which does really well with the training process. So here you have two losses, and in order to speed up the training time and to make it not only faster but faster to converge, but also more robust. In the first part of the training process, we take into account only loss coming from the contrastive learning encoder, so we can set its weights correctly, or the way that we want, and we include the loss from classification. So this jump here, you can see it's the moment in the training process where we introduced also loss from the classification. So on x-axis, you can see it during the training process. So this would be the methodology. And now when we obtain the notations, we need to test them to see whether they make sense. And the current problem with this entire area is that we don't have gold standard reference benchmark data sets. So it's still a young area, and it's not like in the DNA sequencing where you have multiple samples with very accurately reconstructed variants. Also in the somatic variant calling, nowadays you have high quality gold standard reference data sets, but here you don't have that. And you need to find some mechanism or some matrix to compare against the existing solutions. And in our work, we propose two matrices. The first one is performing comparison in the cell type annotation process on subsampled data sets. So we have single cell reference data set with a high quality, and we perform its downsampling with different factors shown here on x-axis. And we use this downsample data set as a test. So we annotate it, and we already know which are annotations because they are the same like in the single cell. And we tested it against different tools that are available and mostly used for cell type annotation with the reference, and we managed to obtain the highest accuracy on almost all subsampling factors. As a second matrix, we proposed the retention of the marker genes. So in the single cell data set, which is high quality, you have cell types. So cells are grouped, and inside of each cell type, you can calculate these marker genes. And in the spatial transcriptomics data, in the same cell type that we assign, we want to have same marker genes, or at least as close as possible. We want to match them. And this matrix, it measures the preservation of the marker genes in the obtained cell types. So these box plots, they show for each cell type how many, which percentage of the marker genes was preserved in the annotated spatial transcriptomics data set, and also in majority of the different samples we were able to have the highest retention. And this would be the final result. So we have a data set with annotated cell types. How much time do I have left?
You have 6 more minutes.
Oh,
okay, 5 more minutes.
And when we have these cell types annotations, what can be done with it next? So we want to get more insights. We want to identify some functional regions of interest which can be connected to some structural change or to some function of that tissue. So here we decided to use this mixture of the cell neighborhood that is present. As you can see here in this sample, you have large mixtures. So different cell types exist on the very close one to another, and since they do that, they probably do some common function together. And based on that cell neighborhood, we decided to identify these cell communities. How we did that? So we divided the tissue into these small windows, 1, 2, 3, 4, and inside of each window we look percentage of each cell type in it. And we write down this percentage as a vector, this percentage is vectors, and we do it for all the windows in the tissue, and we can perform clustering. So every cluster would represent a community. We can also do it with different sliding steps in this window and perform clustering on it. We can do it across different slices, so we can process together many different samples. We can do it with different window sizes, and as a result, we have obtained these communities which might represent some functional regions. So for this very well explored tissue of the mouse embryo brain from day 16, we looked at the existing databases such as Alan's brain, and we found matches for this central orange community. We matched it with the dorsal tire of thalamus in this brain arc. The brown community, it matches with the mental zone of dorsal pallium. Next, we analyzed these 3 liver cancer samples. One represents healthy liver with a huge amount of hepatocytes, as it should have. This one is a borderline, and this is a tissue invaded by tumor. And as a result of the cell community detection, we managed to detect this green borderline community, which has a percentage of hepatocytes which is between 40 and 60%. And we can actually see the regions where the tumor is evading the rest of the tissue. Both of these tools are available in DCS Cloud platform, and they are also available on GitHub. We submitted those 2 papers in the journal, still waiting for the response. I would like to thank the organizers for giving me the opportunity to present this. I would like to thank our team here in Serbia for striving together towards our common goal, which is omics for all. And thank you for your attention.
We have time for one question.
So, thank you for the presentation. I wanted to ask, what is the process to get those samples? So, for example, mouse kidney has this 3D structure. You showed that, so it has like layers of cells. So, do you have to like slice?
Yeah, you slice it out in a very thin way with some hard knives, and you can slice it literally in the size, which is size of the cell, like 10 micrometers, and you put that very thin slice on the chip, and you perform the process.
I just thought that maybe it exists some kind of instrument like MRI to get those samples.
Yeah, there is an instrument to perform slicing.
Ah,
Maybe one more brief one.
Yeah, I wanted to ask a question.
Yes.
If anybody else wants. So, thanks for the talk. I think it's quite interesting in general, this approach with the contrastive supervised learning. So, just wondering if you were just trained for the supervised loss without contrastive learning, right? What's the impact on accuracy for the classification nodes? Did you also get benefits in classification by adding the contrastive part? Or, I mean, if you were just train the model to classify, maybe you would get something similar. So, do you get large benefits from the contrastive loss part for the classification task alone? If you get
Yes, so we have huge number of features. So we have like 30-40,000 of genes, and it's not a good practice to. I mean, you cannot pass all of these features to a classifier. It would actually have maybe smaller number of samples than features, so it doesn't make sense to perform the process. And with the contrastive learning, we perform compression, we perform dimensionality reduction, and we obtain, instead of 40,000, we obtain 256 or 100 features which can be passed to the classifier.
How many classes do you have there?
Classes depending on the tissue, but let's say from 10 to 50.
Okay. I'm asking because you could still put the encoder part, like run through all of that, but then just classify without the contrastive loss. So.
You mean to lock it?
Yeah.
Yeah, we experimented with different different parameters, and we experimented with also having to allow smaller change in the contrastive learning part, and these are all parameters that can be adjusted in the tool. Thank you.
Okay. Thank you, Vladimir. I have your certificate here, and thank you for joining us. Can we have